Thomas Hunt Morgan. Thomas Morgan used the fruit fly, Drosophila melanogaster, for his experiments, which paved path to reveal both the phenomena, linkage and crossing over. Drosophila melanogaster, the fruit fly. Gray body of fruit flies is dominant over the black body. Normal wings is dominant over vestigial wings. Morgan crossed homozygous gray-bodied, normal-winged fruit flies with homozygous, black-bodied, vestigial-winged flies. All flies of F1 generation were gray-bodied, normal-winged. The result is similar to the result that Mendel got for his dihybrid cross. Then, Morgan did the test cross for F1 members. But he didn't get the results that Mendel got for a test cross done for a double heterozygote. Mendel got 4 phenotypes, with 1:1:1:1 ratio, for a test cross done for a double heterozygote, but Morgan didn't get such a ratio for the test cross, though he followed the same procedure. He got 965 gray-bodied normal-winged flies, 944 black-bodied, vestigial-winged flies, 206 gray-bodied, vestigial-winged flies, 185 black-bodied, normal-winged flies. Morgan's results as a percentage. Gray-bodied normal-winged flies 41.5. Black-bodied, vestigial-winged flies 41.5. Gray-bodied, vestigial-winged flies 8.5. Black-bodied normal-winged flies 8.5. Mendel's results. Morgan's results. Then Morgan assumed that the 2 genes for body color and length of the wing should be located on the same chromosome as linked genes, and he explained that the alleles of the linked genes cannot be segregated independently, they enter to a gamete together. Let's observe the flies resulted when these gametes fertilized. Gray-bodied, normal-winged flies. Black-bodied, vestigial-winged flies. Random fertilization of gametes of linked genes. Gray-bodied normal-winged flies. Obtained results are the first cross, resembles the expected results. Let's observe the flies that can be resulted by the test cross. Gray-bodied, normal-winged flies. Black-bodied, vestigial-winged flies. Gametes of linked genes. 2 phenotypes should be resulted, with 1:1 ratio, when these gametes are fertilized. But The obtained results of the test cross done by Morgan did not resemble the expected results. Then Morgan introduced another phenomena called crossing over to explain his results. In gametogenesis, parts of non-sister chromatids of pairs of homologous chromosomes are exchanged due to a phenomenon called crossing over. It takes place in prophase 1 of meiosis 1. Crossing over, which takes place between of non-sister chromatids. Gametes produced by crossing over. Nature of gametes produced by crossing over. Let's see the result that can be obtained when these gametes are fertilized. Gray-bodied, normal-winged flies. Black-bodied vestigial-winged flies. Gamete formation, with crossing over in heterozygote F1 flies. Gamete formation, in homozygous recessive flies, used for the test cross. Random fertilization of gametes. Gray-bodied, normal-winged flies. Gray-bodied, vestigial-winged flies. Black-bodied, normal-winged flies. Black-bodied, vestigial-winged flies. Parental types. Two new phenotypes are resulted in addition to the parental types of flies when crossing over takes place. New phenotypes arise, are called recombinants. Gray-bodied vestigial-winged flies and black-bodied normal-winged flies are recombinants therefore. 4 types of flies are seen to be produced in similar numbers this way when crossing over takes place. According to the Morgan's results, each recombinant type has been obtained with 8.5%. Since the ratio of 4 phenotypes with crossing over is similar, percentage of each phenotype should be 8.5. But the percentage of 4 types of flies obtained in Morgan's crosses do not resemble these percentages too. Morgan got more parental types and lesser recombinants in his test crosses. Let's try to clarify how Morgan got those numbers by analyzing the data in the process further. We considered the phenomenon crossing over and the flies resulted due to crossing over so far. But crossing over is a random process and it can be taken place between some pairs of homologous chromosomes only. It does not take place in some homologous chromosomes therefore. Let's pay our attention towards the gametes produced without crossing over, and the flies produced by their fertilization, to clarify the Morgan's results. Number of each type of flies resulted in Morgan's crosses: 965, 944, 206, 185. Recombination frequency can be calculated by these numbers. We can find out the percentage of chromosomes which do not undergo crossing over in gametogenesis by calculating the recombinant frequency. According to the data of this cross, the recombinant frequency equals 17. Recombinant frequency resembles crossover frequency too. Crossing over takes place in 17% of chromosomes in gametogenesis, since recombinant frequency equals 17 of this example. Crossing over does not take place in 83% of chromosomes in gametogenesis therefore. Let's see the type of flies produced without crossing over, the types of flies produced without crossing over. Gamete formation without crossing over. Random fertilization of gametes of linked genes. only parental types of flies are resulted, when crossing over does not take place. Now it's clear that parental types of flies are produced in both the instances, with crossing over, as well as without crossing over. Accordingly, flies produced with crossing over flies produced without crossing over. Total. Recombinants are lesser in number, since they are produced only with crossing over. Now it's clear that Morgan has got more parental types of flies, since they are produced in both the instances, with crossing over as well as without crossing over. The summary. More parental types are produced, since they are produced in both the instances, that is, with crossing over as well as without crossing over. Recombinants are lesser in number, because they are produced if crossing over took place only. Significance of Morgan's experiments. By conducting statistical studies about fruit flies, Thomas Hunt Morgan came to understand that genes are arranged in a long row inside chromosomes and discovered the crossover phenomena. His experiments on the fruit flies could confirm the Mendelian inheritance and the chromosome theory introduced by Boveri and Sutton. Thomas Hunt Morgan won the Nobel Prize for Physiology in 1933. In 1902, Walter Sutton and Theodore Boveri developed the chromosomal theory of inheritance. Boveri and Sutton's chromosome theory of inheritance states that genes are located on specific sites of chromosomes. More related facts: the distance between linked genes can be calculated with all these knowledge. Recombination frequency resembles the distance between two linked genes. It's calculated by map units. As an example, if we consider the two linked genes that determine the body color and the wing length of fruit flies, we discussed here, are located 17 map units apart. Thank you for watching. Subscribe for more such videos.